That's always good. Yeah.
133.
That's really funny. I think I'm gonna speed.
Yeah.
Is a speed walk. So I have to speed walk. Oh, no, it's just one of those natural things.
That's quite numerous, honestly. It's like exerting. It's silly goofy. It's silly.
Hey, gang. Hey, good gang. Never mind. Okay.
Actually, I should call myself isolating because I think I'm most productive when I self-isolate. Except sometimes I'm not productive when I self-isolate.
All right, guys, this is started.
That's right.
So, I have a few good questions coming in from the little cards you guys did last time. One of the ones that kind of asked a little bit, I was talking about kind of going over a little bit again over the DNA, the proteins, kind of how we're making cables and how these cables basically work together through different paths. So today we have a pretty easy lab. Class, we're gonna be talking about just the cell. So the big thing about today is we're just comparing the prokaryotes and eukaryotes, and then a little bit of comparison of within the eukaryote plant. We're gonna go over a few organelles, a few of their jobs. That'll be the main thing. Afterwards, I'll talk a little bit about microscopes, but we'll have like a 20-30-minute gap at the end. Where I'll see if you guys want, I'll basically like field your questions about the beginning protein. Yeah? All right. All right, so to start off, again, same thing. We'll go heavy in the first few minutes. So this chart looks kind of pretty heavy and it has a lot of material on it. But there's only 4 things I really want you guys to remember here. It's going to be prokaryotes, right? That's going to be looking at really small cells. They're kind of boring. They don't have that many organelles. Then we have the fun ones, which is the eukaryotes on the right. And then these basically have a lot of organelles inside of them. The cool thing about the organelles is they're each like its own little chamber, like surrounded by a phospholipid bilayer. So it's a little bit of like a little fat bubble that can seal that environment to do whatever you want within that little chamber. So we're going to go today through all these little chambers, kind of talking about how is that chamber special, what kind of jobs does it accomplish. So that's what your eukaryote stuff. When we get into this one, the two that we're going to go over a lot in the class are going to be animal cells and then plant cells. Right? So as long as you remember that, eukaryotes versus eukaryotes, within eukaryotes, animals and plants will be okay for the purpose of this class. Yeah. All right, so the first big thing we're going to talk about between prokaryotes and eukaryotes is prokaryotes themselves are normally going to be unicellular organisms. That means they only have one cell. That cell is then responsible for doing everything, right? So it's got to be able to breathe, it's got to be able to eat, it's got to be able to go to the restroom, it's got to be able to reproduce, right? This thing's going to carry on and do all those tasks, and it's kind of on its own. When it comes to eukaryotes, we have some eukaryotes that are unicellular, but we also get into these fancy things now where we're like multicellular organisms. The cool thing about that one is now that we have these different cells, even a simple thing like diffusing something in, it's very easy for a single cell, right? Has all the surface area stuff to come in. For us, it's like I can't absorb oxygen through my skin to be faster, right? So for this, we end up having a lot of different cell types, and in each cells, they'll specialize. So they'll make specific proteins, they'll change their shapes, they'll change even how they interact with each other to be able to fulfill a specific task, right? Like we could talk about, for example, our lungs. The cells there will end up giving themselves like these small vein patterns where there's like little branches on branches on branches on branches. And the whole point of that is it gives us a huge surface area. Same thing with our like stomach lining. So our gut has like little curvatures like that as a tissue. And then if you go and zoom into every single cell, it looks like it has a bunch of fingers sticking out like this. And every finger that has a protrusion is basically this thing has more surface area. So it means that even though we have a thin gut, it has a thin surface area of stuff that it can eat, right? So yeah, we can go through all this stuff, but the main thing is multicellular. The cells are going to then pick different tasks, and then each one of them is going to be able to carry on different responsibilities, whether it's breathing, eating, going to the bathroom, and then of course reproduction, right? All right, so the thing that we're going to find in both camps, whether you're talking about prokaryotes or eukaryotes, is going to be these things, right? So the first is the plasma membrane. So that one, I feel like you guys are probably tired of me talking about phospholipids already, but we'll do it one more time, right? So the first thing it's going to give us is going to be this thing where you have the phospholipid bilayer. On one side, we're going to have the head, which is polar, right? So this is going to want to play with water. Then we have the tails like this. And here we're looking at tails, non-polar, and then these do not like water, right? So this is going to be like, I'm not going to play with water. This thing is one of the biggest, critical, most powerful things that we got, because what's going to happen is if any molecule is trying to come into our cells, so coming from the top, and this is the inside of our cell, if it's polar, it's cool. It'll interact with the purple side, right? And they'll hang out with them. They'll be able to grip each other. But that polar molecule will not want to interact with the tail. So that's going to be a polar molecule will come here, get stuck, and it won't be able to make it. On the other hand, if you bring a molecule that's non-polar, you get it to the surface, you're going to have to jam it into this middle layer. Once it's in here, it's happy interacting with all these carbon, hydrogen, non-polar stuff, because they use those van der Waals, so they'll be super happy. Problem is on this other side, inside your cell, you also have water. So that thing's not going to want to be. So it's either non-polar or polar. This thing's going to be the big thing that's going to trap you, right? So again, this is the big foundation that they all have. It's the big protector that on the inside of this is the living organism. Outside of this is the environment. So within that, so let's say this is the inside of the cell. This is where we have our cytosol, right? So in this case, it's very, very close to the environment on the outside, but there'll be some differences. And I want you guys to think that this is like, you basically have water, right? So we have our little fat bubble. Inside of our fat bubble, our cytosol is going to be where we have like the water inside of our own cells. Right? Then both prokaryotes and eukaryotes, they both have DNA, but we'll go today how they hold them in different ways. Prokaryotes, because they're super basic, they just have them randomly in the cell. Or eukaryotes, we're super fancy, we got them in a little special compartment, which is the nucleus. Right? And then the other big task that we all have to accomplish is going to be working with ribosomes. So ribosomes are the cool ones because remember, our DNA gets transcribed, and makes RNA, or RNA gets translated by the ribosome, and that's what actually makes the amino acids, like our protein chains. Right? We both have them, and like again, we'll see that they're basic. The ribosomes are just free floating in the cytoplasm. We have cooler traits or fancier stuff we can do with ours. All right, so let's look at this picture. Sorry for flicking on you guys. I'm putting the prokaryotes on the right. So when we look at the cell, this thing isn't actually drawn to scale. Prokaryotes normally are super small. They're way smaller than our eukaryotic cells. But the thing we're going to look at is because they don't have any extra organelles, like their DNA, you can see it's just floating around. And this is basically their plasma membrane. Right? So at that outer fat bubble, our DNA is basically just hanging around on the inside. In comparison to the eukaryotic cell, which is, again, the fancy one with the organelles, and then you have this thing. So if you look at the blue region, like this outer line, that basically represents a phospholipid bilayer. So again, we have another really cool shield like this, right? Hydrophobic, hydrophilic, which is going to help us block things. We can control what comes in and out because it has little pores. But we can basically hold our DNA in that environment. And to be honest, that environment is made to be safer, to keep our DNA healthy, like it protects it better. The next level, we're talking about a little bit of the organelles for this. This is gonna sound a little convoluted, like it's gonna be mixed into each other, but this is the way that I kind of really, it really helps me remember. So story-wise, the organelles I'm really gonna have you guys focus on is gonna be versus your nucleus. Right? And then the big thing about this is the one that's gonna be holding on to your DNA. Right? From here, DNA gets transcribed, transcription, and this is the one that's going to go ahead and produce our RNA. Right? That RNA is then able to come out of the nucleus because it's a smaller molecule, and it can go to the ER. So this is the next big environment. Nucleus, and get ourselves to the endoplasmic reticulum. Once it gets to the ER, one of the jobs for the ER is that when you look at it through a microscope, there'll be a part that looks smooth with these little curves, and some of them look, they call it rough. We have all these little bumps. And those little bumps are basically our ribosomes kind of hanging on to that phospholipid bilayer, right? So in that case, ER is the environment. We have our ribosomes that are going to be helping us out. And this gets us to the next step of that RNA is then turned into our protein. So at which point, started with DNA in the nucleus, in the ER, nucleus, we made the RNA, that got sent out to the ER. In the ER, we've made our protein. Now that we have our protein, the next step is going to be, we're going to work with the Golgi complex. So the Golgi complex, it does it in a fancy way where it grabs the proteins and it glues sugars onto it. And based on how it glues the sugars or it does the modifications, these things able to send them to different compartments. So you can say, hey, I want some proteins to get to the plasma membrane. They'll go through here, the Golgi grabs them, throws them away. You can also send them to like the mitochondria, it can send stuff to the lysosome, right? So this thing's kind of like the post office of the cell. Once you've made the protein, this can go and distribute it to wherever it needs to go. And then the last organelle, which is going to give us kind of like the storyline, is going to be this guy, the lysosome. So imagine kind of like a gene has a life. It goes DNA, RNA, protein. This thing will send it to the compartment. It does its job, and once we're done with it, either it's completed its job, it's getting old, and we want that thing off, we'll send that protein to a lysosome. And what the lysosome does is, again, it's one of these organelles, so it's a little fat bubble, and on the inside, we balance proteins that love to cut other proteins, but they love to do it in an acidic environment. So normally the cool trick about this guy is, we'll bring a bunch of acids, bring a lot of those protein cutters, and then whenever we don't want a protein anymore, we'll throw it into that compartment and just clean it up. So, kind of going back to yesterday's lecture, Monday's lecture, it'd be like, in here, the ribosome, right, is doing the dehydration reaction, where it's gluing all the amino acids to make my long cable. In the lysosome, we're doing the opposite, right? Bringing in water, breaking the chain, and we reduce the amino acids. The cool thing is, because we break them in those little subunits, technically we dump them back into the cytoplasm, and then you can reuse them again, because they're back to being like at their base state, or like a monomer state. All right, so organelles, nucleus, ER, Golgi, lysosome. Everyone feel pretty about those? Yeah, so this is the ones that I really want you guys to feel comfortable with. This thing's messy. It does everything in the whole cytoplasm. So it'll be making things, making RNA. The RNA goes, hits the ribosome. It'll be making protein already in this environment. And that's because it doesn't have anything like we do, where we have to do really fancy tricks to be able to get something inside of a plasma membrane, throw it into another organelle. So we'll go a little bit into it. In this class, in 233, I'll go into it heavy because that's where I can really be free. All right, so here's a little bit of a chart of us bringing in together all the stuff. We went over this in the beginning, right? Things that both of them are going to have. And then I want you guys to jump into these outer parts, which is prokaryotes have some things, eukaryotes have other things, right? The big thing there is organelles. It's going to be the big player, right? All right, and here's my little list. this is how I remember it. Because to me, I like to think about everything's kind of central dogma, which is the storyline of how does the gene get made and how do we throw it away. Right? If you're not a storytelling person, but you're a visual person or you like the list, then basically work with this guy, right? But it's the same information. Nucleus, that's the one that holds our DNA, our ER, protein synthesis. So again, because the ribosomes are there, they'll be making a protein. Our mitochondria, which is these guys here, they're basically where we make energy in our cells. Like we literally break our sugars in this thing and we can actually get the ATP out, which is the power that we need. Again, the Golgi apparatus, that's where you do the packing, or packing and sorting, because you can send stuff out. And the last one is your lysosome, where we're getting rid of that protein stuff. Everyone feeling pretty good? Yeah? All right. So now that we're pretty good with eukaryotes, the next step is within that, we're really going to focus basically on these two groups, which are going to be animal cells and plant cells. So everything on the right here is really just showing off what are the things that we do the same, right? So if we do it in my favorite way, which is telling the story, we start at the nucleus, right? Start at the nucleus, this little purple structure. The purple actually represents the phospholipid bilayer, so it's basically this thing. And there here you have your DNA, and there's your DNA, right? Afterwards, you move out. There's your ER here in purple, all these little grooves. Here's your ER. This is the place where we're going to be making lipids. The rough parts will be making our proteins. Then our proteins will make it to the Golgi, there and there. And again, remember, that's the place where we're going to sort our stuff and we'll send it to wherever we need to. And then here's one of the big differences, right? So the little stuff on the left. For animal cells, we basically use a lysosome. So this will be, again, the place where we're going to break things open, cut up the DNA, we can cut up proteins, whatever we need. That's special to animals. The other one is this little structure called centrioles. And these are pretty cool because what the cell does is when it's time to divide, it first puffs up, right? And then it has to physically grab half of the DNA for itself, and the other daughter has to grab her half of the DNA, and they have to pull each other apart. So then those things are going to be the cables that build like this little scaffold, like little, almost like wires that they push off each other so the two cells can walk away. And at the same time, they leave other cables behind that grab onto the DNA and they put it towards the. So for cell division, these are super cool. I'll show you some data that it actually looks pretty fun, but once we get closer to the DNA stuff. So special animals, now we jump over here to the special to plants. So the first one that we're going to talk about is going to be the cell wall. So for this, we have the plasma membrane, the little white line. And that's this thing, right, where we have all the molecules lined up, and they're really being held in place just because we have van der Waal helping out here, and because these only want to work with water, and these only want to work with water, that's why they don't pop out. So strength-wise, that's pretty weak, right? Like it's not really holding on to itself. So in this case, like if I took some of your blood and I just changed the water, the amount of ions in water, if I get like pure distilled water and put it into your blood cells will instantly put in so much water, they'll pop, right? Because that grip isn't that really good at keeping this thing in one piece. This thing has this thing called the cell wall, which is basically built up of sugar cables, like literally little 6 rings glued onto themselves to make one cable. And that would be one strand that coils into other strands to make a bigger cable. And then those cables will mesh into each other. So it ends up being like, if you imagine we're a balloon and we can inflate it and puff it up, this is like if we have a balloon and then we tape it all over the top. Right? And that would give so much mechanical strength where I can put it into water and these cells won't pop because they have so much protection, it's not gonna make their membranes kind of burst. So, big thing that sets them apart, cell wall. The other one, the one we really care about, is basically this guy, the chloroplast. So for the chloroplast, you're looking at this little thing right here. This one's pretty cool because it has 2 layers of this. Right? So this outer green part is going to be our first compartment, and within that there's these little structures that have another set of those little membranes. And what we're doing in that one is, when light comes in, it hits a set of proteins, and those proteins literally activate electrons. those charged up electrons are the ones that we use to help catalyze the reaction of gluing the carbon together to make a sugar. And that's how we can go from sun energy, we turn it into chemical energy by messing with the electrons, and we store it inside that sugar molecule. And then we grab that high energy sugar molecule, bring it into our mitochondria, and then we tear it apart. And as we tear it apart, we're basically charging ourselves. So super important. If we lose this, we technically wouldn't be able to survive at all. Or in any species, we lost it in general, we'd be done. The last one, I'm not gonna bring this one up that much, but it's the vacuum. So it's this little pocket, and it's basically like a water-filled cavity, and they can kind of control it. They can make it bigger. They can also squeeze stuff out, and that's one of the ways they can also help themselves regulate the pressure. But for the class, these two are my favorite. This one, I would say remember both of them. That'd be pretty cool. All right. So that was the heavy part. Yes.
What's the difference between the smooth and the rough yarn?
Oh yeah. So it's just let me see if I can find your picture. All right, we're gonna look at the picture first, and then I'm gonna go back and I'm gonna find you a video just so you can see how cool it looks. All right. So if we're looking at this picture, here's your nucleus, right there's your DNA. This will be a phospholipid bilayer on this side and on that side. Right? So it'd be, if I drew this in like my quick mode, it would look like that. Here's my two tails, and then these would be the different heads, right? That's what we're building our wall. What's gonna happen is our ribosomes, in some spots, they'll be building up right here with these other proteins that kind of make a channel to this. And then as the RNA is being read through the top, we're making the amino acids go into the cell or into the ER, so we'll pump them in. So there'll be regions where there's a bunch of these ER molecules and you have these transmembrane stuff. And that's when you'll see all the bumps. So the ribosomes are the thing that cause the bump to happen. And then on the other side where we have the smooth one, that's where the cell's normally working on making lipids. So it's actually helping to make these components. Right? And what happens there is that very often, you'll have this thing where it buds off. So you'll have, let's say, your smooth membrane set up like that. And this cell's gonna be making lipids and kind of jamming them into this thing, right? So we'll be adding more molecules, making this a little bit longer, making a little bit more, longer, and then more. And then eventually it makes it like a fat bubble and it'll throw it away to the next compartment. At which point this thing does this cool thing where it looks like this. That'll curve up. This inside parts will kind of go like this. This one will curve all the way like that for a second. Right, so it'll kind of pinch off, and then eventually this gets clipped. And then this will seal on itself. And this will seal back to the original, something like that. That drawing's a little rough, right? If you guys want, give me 1 second, let's look at a quick video, and then you'll see what I'm talking about. Smooth ER creating or budding vesicle. Come on, buddy. So it's actually super fancy. So in yellow, we're basically looking at the phospholipid bilayer. You see kind of how it's puffing itself up. It has ways of recognizing that it's grabbed onto the lipids. It'll pull it together and pull that out. So what ends up happening is these things sneak in there, they'll kind of get kicked out. So you'll have regions where there's no ribosome just because you're throwing out these little fat bubbles to replace lipids. And then they'll build up in other regions where they're actually just making protein and stuff. Does that make sense? Yeah? Good question. Any other questions?
Could you go back to the slide that had the organelles and their functions next to them?
The really short ones? This guy?
Yeah, that one. Thank you.
So in the book, when you guys are reading it, they're gonna give you longer definitions. They'll also give you more inclusive definitions. For the sake of my test, just remember these and we'll be fine, right? Like stuff, I'm not gonna force you, it's like the ER, that one, because so if you look inside of that one, inside of here, it actually has its own DNA. Like it has its own little small genome. So those are chloroplasts where photosynthesis happens, they have their own DNA. So in your book, when you read it, you're gonna say, mitochondria, energy production, / it contains DNA, it contains some DNA. But just stick with mine, we'll be okay. Same thing with the Golgi. In the book, they'll tell you the Golgi does a lot of other things because it does help like consolidation, it does all those modifications, it also helps the lipids. But for the sake of our storyline, just stick with this and we'll be all right. You know? Any other questions? No? Andrew? No. All right, if that's the case, this is where we get to nerd out a little bit. For this one, I'd like to talk a little bit about microscopy. I'm gonna go over it pretty fast, but the main thing I want you guys just to know is that when we're looking at the stuff we're looking at now, we're super zoomed in, right? We literally have just jumped up from atoms to molecules to our first looking at cells. All of that, those things you can't see with the naked eye, right? Like the wavelength of like red or something, I think it's like 600 nanometers, which is somewhere around this scale. So the color red vibrates at that wavelength. That thing is so big that technically we can't be used to image none of this stuff down here. Because if you're trying to take a picture of it, your light will literally kind of bounce around it and it won't like be able to hit it. So this is where we have the most zoomed in microscopy we have right now, which is that electron microscopy. So instead of shooting light at things, we're actually shooting electrons. And what's going to happen is you can set them up in two different ways. So in one way, it's like imagine I'm the electron producer and I'm trying to take a picture of the class. What it's doing is it'll shoot electrons down like that. And then in this case, I have a detector off to the side. So it'll be like, if I throw something and someone's there, it'll bounce back and I'll pick up a signal that pixel, it's either there's something there or there isn't. So as I'm going kind of like this, what it ends up doing is it picks up what's on the surface, right? So here, I'd be able to, my electron would hit Irani, but I wouldn't be able to hit Colin with this electron. So I would get a picture of her, now him, as we're going back down. Same thing here, because I got a good shot, I would be able to shoot all the way back. I would hit Andrew and know that there's a person there. And that's how this one's basically working, right? So I can't remember, I think these are spores or I think, I can't remember this, these might be spike proteins. These are the coronavirus vibes, right? So this would be the things on the surface of that cell, kind of holding it together. And this is the same trick, right? We're shooting electrons. If they bounce back, we know something's there. When the electron doesn't hit us back, then we think it's a dark spot. Right? And just with that, we can actually get images of the surface. The other cool one is this TEM, which is on this side, which is transmission microscopy. In this case, you have to grab all your samples and you slice them very, very thin. And what you're doing with the slices now is the emitters on one side and then the detectors on the other side. So now it'd be like if I'm sending the signals and then Colin's picking them up, right? So it'd be like if there's nobody in the way, the signal goes through, he gets the signal, that makes it white. But if something's in the way, whenever I'm at this spot on the certain signal, he's not going to get any image. So that's when we get the dark stuff. So here you can see they're kind of going through the cell. There's your phospholipid bilayer. This would be this outer head. This would be the tails here, and then that's the inner heads. Right? They're super high density. That's where you get a nice dark spot. Here you can see this is the edge of your nucleus, and there's your nucleolus. There you have like your DNA is like super coiled into itself. So it's high density. So no matter how much particles you shoot, it's hard to kind of image through it. But in this case, you can actually look through samples. There's a really cool one now that I was looking at what you do is they'll grab these and they'll slice them every like 8 to 10 microns. So you make super thin slices and you just image all the slices and you can put them back into 3D. And you can look at 3D images of like super fun cells or super fun organelles. So super powerful stuff. The bad part about this is because it's electrons, you have no color. Right? So everything we look at is either there's more signal or there's less signal. But you really can't tell stuff apart. That's where we go back to. So again, electron microscopy, we have the best resolution, we can see more things, but everything's black and white, right? Next, we're going to jump into this thing called light microscopy. So for this one, now we are using light, and then we can use, you guys probably seen stuff like this. Right? So this would be the one where you just are throwing white light, and then depending on what your sample is in the, like whatever sample you put in between you and the light, that's what your eye will pick up. You multiply it, I think here it's like 110, so it'll be 1,000 times bigger than what it really looks like. For that one, we're looking at regular cells are kind of boring. Like if we hadn't stained this thing would almost look all the same color, right? But what we've done is we found different chemicals that bind to different things. Like one, I promise you, you're gonna hear it over and over again, it's called DAPI. DAPI sticks to DNA. And it basically makes it shine super blue. So it's really cool if you're trying to study cells or if you're trying to study like the mitochondria or the chloroplast, you can hit them with a little bit of this dye, it'll wash into your cells, bind to the thing that it cares about, and everything else will be dark. And then what we just do is we just bring in different colored dyes. So there's DAPI getting us the nucleus. Here, this is one of the cable proteins that kind of build our cells. We've tagged it with GFP. And then we can get some really nice shots. So super powerful benefits is we have a lot of color. The only problem is it's not as good as that one, right? So here we're able to see a cell, like that's our resolution. That over there are proteins hanging out at the edge of the cell. Right? So then this whole thing we're looking at here, this is the phospholipid bilayer, and that imagine it's the curvature of the whole cell. So super zoom, yeah. And then those colorful ones we normally we Photoshop them. I'm doing that. So yeah, those look pretty cool. All right. Then the thing that we used to look at, the biggest things, basically just our eyes there. We can see some stuff like frog eggs. Right now we're working with sea urchins, I think right about here. We really can't see those unless you have a microscope. So this is normally take care of a bigger stuff. So long story short, electron microscopes, you can super zoom in, but it's only black and white because it's based off of density. Light microscope, you have this middle area. You can't look down here. You can't look at bigger things. But the cool thing is we do have color, and of course, you got a eye. You guys on that one, right? You have a pretty good zoom, and you can see. For the most part, we can all see color, right? All right, pretty good. Any questions on this stuff? No. All right, so here it'll be if you guys want to stay, you can stay. If you don't want to stay, you don't have to stay. I'm gonna go over the protein and amino acid stuff real quick. Everyone staying? All right, let's do it. All right, so this one. We're talking information wise. First, I'm just gonna talk about how they work as cables, right? So in this case, here we're looking at the DNA, and the way that we orient ourselves is based off of the sugar ring, right? So when you're looking at this one. For that one, it depends if they draw it this way or they draw it upside down. The easiest way for me to find them is I go look for wherever the our amino base is at, right? So CAGC, and that's spot number 1. And you just count in clockwise pattern. Right? So 1, 2, 3, 4, and 5. And now I know this is the 5' phosphate, and this is the 3' hydroxyl. Right? So in my head, when I'm thinking about these, my super simple version is at the 5', you go to the P group, you go to some carbons, and then it goes to an OH group. Right? And this will be a 3' side. When the next DNA molecule comes in, it's going to bring in its phosphate, its little carbon chain, and then the OH group. And then it keeps doing it over and over, right? But every single time, what we see is 5' is this side, 3' is on that side. So DNA, RNA, this is the way to say, the start. It will be 5 prime, and then the end of the sequence, and then we call that 3 prime. So, just cable wise. Let me jump back real quick. So this is the best slide I really prefer to look at when we're talking about how we build the cable. So this R group is where we have all the fancy stuff, where it's specialized. But this cable back here is a thing that doesn't change, basically that's the same thing over and over again. And just like this one, instead of starting with a phosphate, it starts with a H3N. Right? So you can see the nitrogen group. That's what you're focusing on right there. And that's what we call this. It starts with the N terminus. And what you're going to see is N, we hit a carbon, right? Then we hit the other carbon, and this one's C double bonded up, C double O, right? So N terminus, that's going to be our C terminus. When the next amino acid comes in, it's going to come in the same way, right? It's H3N, the little chain, and then we're going to glue in that CO. This gets put together, but as a cable, we still have N at the front and then C at the back. Yeah. So when you're following the cable, just keep an eye on these. H to C, H to C. Now, after we've linked it up, H to C, sorry, N to C, N to C. Does that make sense? Yeah. Pretty good? All right. So in that case, these are the two parts of both cables that are repetitive. There's not going to be anything really special about these guys. There's nothing going to be to tell them apart. So now let's go back to about how do we actually tell these guys apart, right? So for this, in this case, this is where we can add the different, so once you have your little sugar ring, spot number 1, this is where we're going to add the nitrogenous base. There, you guys have 4 options. For DNA, you can make it be A and T, or C and G, right? This is the combos that you can put in. Whenever you decide to pop into each one of these spots, are basically going to tell us what we're going to be working with, right? So if we go look at by this little picture looks a little bit complicated, but again, here's our cable, right? Yellow, blue, yellow, yellow, blue. Here's our nitrogenous bases. These little black dots are representing hydroxyl groups. So again, those are going to be polar interactions that are hanging out there that are going to want to grip other polar interactions, right? These will make it so that they match up with a specific pair, but all the empty ones that are left open are basically spots where you can have other things come in and interact with. If you switch from an A to a T, you can see here there's no hydroxyl group, but there you're going to have one there. So you're going to have a polar meeting, and just these little differences are going to set these guys apart. You don't have to go into detail about what the differences are. The only thing I want you guys to learn from the letters is A pairs with P, C pairs with G, and then the whole 2 rings versus 1 ring. Yeah. And as you can imagine, 2 ring versus 1 ring, it'll be, are the hydrogen bonds closer to one cable or are they closer to the other? If I flip this sequence, all this stuff will basically be shifted this way. Right? And that's going to be the special thing of how do we set this molecule apart. Right? Now on the right, if you go chase the protein, this is where we're going to have that R group cube. Right? Like that. Now the R groups for there's 20 amino acids. You've got to remember only 4 types of amino acids, and I'll tell you exactly which ones I'm going to follow. Right? So the main big player there is going to be so I'm going to draw a quick cable, and I'm going to do like an imaginary thing. So let's say I draw this will be my amino N terminus, and there's my cable. and there's my C-terminus, right? If to this, I add R groups that are gonna be non-polar. This thing's gonna have stuff sticking out that doesn't wanna play with water. Right? So if I put any of these chains of amino acids, these little functional groups, doesn't matter which one. These, when you think about them, how they're gonna interact with the plasma membrane is the non-polar ones, if they're all non-polar, they're gonna be happy being jammed right here in the middle, right? Because you're gonna have non-polar stuff working with non-polars. If I change the sequence, and then I pop in this first group, which is gonna be my polar hydrophilic stuff. Again, we can draw my little chain again. There's my N terminus, there's my C terminus, and here I'll be drawing in the polar, right? So it's polar, you know, it has a little plus area, a little minus area, polar, polar, polar. If I'm working with these, we're gonna have that chain of amino acids. If I try forcing it in here, it's not gonna work, right? It's gonna come and bump into non-polar, and it's like they don't mix. It's gonna push it out. But the polar one, because this is polar, it will stick to this top spot, right? So just by switching between non-polar to polar, I can tell it, you can kind of set it up what to work with. Normally when we're trying to study how a protein works, we literally look at the sequence and we'll be like, oh, there's polar stuff in this region. And it could be like, there could be something like this, right? Whenever you see the OHs, we're kind of thinking there's a polar group there. And we're like, all right, if that's really important, we can just switch it back to this, like a CH3, or maybe just an H group. And now it's gonna be from polar, you switch to non-polar, you put it back in the system, and for the most part, if that part's really important, the thing breaks down and it doesn't work anymore. And then you've proven that that amino acid, because it has a polar charge, it determines the interaction, it'll tell it whether it works or not. Yeah. So for the point of the test, when we're bringing up the non-polar things, I will not have you draw them, I will have you identify them, right? Like you should be able to see it and be like, oh, that's a polar group, that's a non-polar group, it's an acid or it's a base. So for that, the ones I want you to ignore are I'm gonna put a temporary drawing. So we're gonna ignore this one for now. You guys can ignore this one. At this point, everything else left on the table, what I want you to tell is, if you guys follow your cables, right? There's your N group, C group, there's your C double bond O. That's our cable side. Everything above is the group side, right? The R group. If you see the little sticks, that means there's a CH there. Otherwise, it'll literally show CH. And that's the biggest bond that I want you guys to memorize going forward. If you see a C and a H, we're thinking right here, non-polar stuff, right? So CH. The moment you see that, I need you to be on the non-polar things. If we go to the next one, here we can ignore this guy because it has an S and I don't want to confuse you guys. If you see, again, there's our cable, you can ignore this for now. If you see OH groups, so OH or O's, in your head instantly, I want you to go, hey, that's a polar molecule. We're going to be working up here, right? So, yeah. And the ones I really love, it's O with C, N with C, N with H's, and P with O's. All of the polar stuff will be the stuff that we're going to hang out up here, right? So for this one, again, look at those numbers. Sorry, look at the OHs or the single OHs or double bond O's, and you know we're on the polar side. It's going to play with water. For the acidic and basic, you can see the oxygens are the ones that are going to be acidic. The nitrogens are going to be the basic. But the biggest key is just that. If you see the minus signs or you see the plus sign, you know what two groups you have. So you don't have to memorize all the amino acids, just memorize the four groups. Because I can say, hey, we have a protein that's non-polar. What do you think would happen if I go and switch them all to being polar amino acids? And how would it affect its ability to interact with the table? Right? And then you have to connect it, you know, oh yeah, look, non-polar, nobody would love to interact because it's non-polar. If you switch those amino acids to polar stuff, it's not gonna want to interact with the tail anymore. If you're super fancy, you can be even bougie, you like, oh, but it will interact with the head. Right? So that's the kind of question they'll ask. It won't be like draw argument, right? I might give you a few of these. When I give them to you, I'm gonna take off all this little squares. So there's just gonna be the actual molecules themselves without the color stuff. So that's why we want you guys to be able to separate, like, how to identify the cable versus that group that gives us the different tasks. And last time what happened is when people saw these, and they were like, oh, there's a plus and a minus, instantly they put everything as acid or bases. Like they'd be like, oh, this is all acidic. All the amino acids, no matter what category, down here, they're gonna have a plus on this front half, a negative on the back of the cable, where you have to focus if the charge beam is gonna be on the R. Yeah? All right, and then I think that's pretty much it for the protein stuff. I think you guys should be pretty good. Any other questions? No? All right, cool. See you guys on Friday.
I was trying to be so. Did you see that? Yeah.
February 14th.
You're so mean.
And then we have a review the Wednesday before.
I tried to ignore you to have any I was like, okay, sure. I love
She was saying maybe we'll leave round two something.
Well, the exam.
They're not set on it.
It's.
AJ has extra time.
Yeah, hold on, happening. Whoa.
Exam.
I'm gonna leave you because you kept messing with my chair. But it was funny. It was funny. It was, but I was also really,
Okay,
really
the exam.
confused.
Really fun.
love
I might-
having a little giggle.
I might get the
So
exam.
you like stalked me and almost like jumped up from the table. I know, I